Definition of biochips. A biochip is a set of diminished microarrays that are placed on a strong substrate that allows many experiments to be executed at the same time to obtain a high throughput in less time. This device contains millions of sensor elements or biosensors. Not like microchips, these are not electronic devices. Each biochip can be considered as a microreactor that can detect and analyze like an enzyme, protein, DNA, biological molecule or antibody. The main function of this chip is to perform hundreds of biological reactions in a few seconds, like decoding genes. For example, biochips are essentially miniaturized laboratories that can perform hundreds or thousands of simultaneous biochemical reactions. Biochips enable researchers to quickly screen large numbers of biological analyses for a variety of purposes, from disease diagnosis to detection of bioterrorism agents. Digital microfluidic biochips have become one of the most promising technologies in many biomedical fields. In a digital microfluidic biochip, a group of cells in the microfluidic array can be configured to work as storage, functional operations, as well as for transporting fluid droplets dynamically. Types of biochips. DNA microarrays, microfluidic chip, protein microarrays, cell arrays. DNA microarray. DNA microarrays or DNA biochip is a set of tiny DNA spots fixed to a strong surface. A researcher utilizes to calculate the expression levels for many genes. Every DNA mark comprises picomoles of genes which are termed as probes. These can be a short segment of a genetic material under high rigidity situations. Usually, probe target hybridization is noticed and counted by recognition of fluoroscope or chemiluminescence labeled targets to decide the relative quantity of nucleic acid series in the target. Microfluidic chip. Microfluidic biochips or lab-on-a-chip are a choice to usual biochemical laboratories and are transforming several applications like DNA analysis, molecular biology procedures, proteomics, which is known as the study of proteins, and diagnostic of diseases. These chips are becoming more complex by using thousands of components, but those components are designed physically, called as bottom-up full custom plan, which is a very large workforce. Protein microarrays. A protein microarrays or protein chip method is used to follow the actions as well as connections of proteins, and to find out their function on a large scale. The main advantage of protein microarrays is that we can track many proteins in parallel. This protein chip comprises of a surface for supporting like microtiter plate or bead, nitrocellulose membrane, the glass slide. These are automated, rapid, economical, very sensitive, consumes less quantity of samples. The first methodology of protein chips was introduced in Antibody Microarrays of Scientific publication in the year 1983. The technology behind this chip was quite easy to develop for DNA microarrays, which have turned into the most generally used microarrays. Cell arrays. The cell arrays accelerate considerably the study of unknown function genes and their potential implications in various diseases by analyzing the phenotypes resulting from an excess or the temporary protein extinction in the small cell islands. This array format seems also particularly interesting for the high-throughput screening of active principles or pharmacological molecules. The manufacture of these cell arrays requires performing opposite and simultaneous transfections of several thousands of different nucleic acids on the chip. Applications of biochips. Pharmacogenomics. Gene expression. Evidence evolution. Biochip as glucose detector. Biochip as oxygen sensor. Pharmacogenomics. The major application of biochip and microarrays technology is in the area of pharmacogenomics. This application is focused in the connection between pharmacology and genomics. The goal of pharmacogenomics is to find links between therapeutic responses to drugs and the genetic profiles of patients. It strives to answer the questions of why some drugs work better in some patients better than in others, and why some drugs may even be highly toxic to certain patients. Pharmacology is the type of science which seeks to understand the preparation, use, and effects of drugs. Genomics, on the other hand, seeks to provide the complete sequence of genomes, and a complete understanding of gene functions and genes themselves. Gene expression. Gene expression is the process whereby a sequence of DNA, the blueprint, directs the production of a protein, the machine that the blueprint describes. All the work in the body is done by proteins. Proteins are the enzymes that digest your food, the cytoskeleton that gives your cells and you shape, the receptors that make your brain function, and the antibodies that protect you from disease. One application of the biochip and microarrays technology is the brain tumor biochip. The BT chip for short is DNA and oligo-DNA microarrays based on differential gene expression in brain tumors. Evidence evolution. The evidence evolution is fully automated random access biochip testing platform. The machine allows any test to perform at any time, as it is a highly versatile analyzer which can operate any workflow. It can receive 2640 tests per hour, which means the machine has a fast turnaround time. As well as this, it allows multiple tests to be performed from a single sample, which reduces time and resources spent on individual tests. The evidence evolution can be tailored to certain laboratory needs and have features including automatic start-up and shutdown, relaxed testing capability, and traceability of biochips. The equipment overall is made to be easy for the client to use, as it includes customizable user settings and easy-to-follow result screens, giving them the fast and accurate operations they would prefer. Biochip as glucose detector. The biochip can be integrated with a glucose detector. The chip will allow diabetics to easily monitor the level of the sugar glucose in their blood. Diabetics currently use a skin prick and a handheld blood test, and then medicate themselves with insulin depending on the result. The system is simple and works well, but the need to draw blood means that most diabetics don't test themselves as often as they should. Although they may get away with this in the short term, in later life those who monitored infrequently suffer from blindness, loss of circulation, and other complications. The solution is more frequent testing, using a less invasive method. The biochip will sit underneath the skin, sense the glucose level, and send the result back out by radio frequency communication. Biochip as oxygen sensor. The biochip can also be integrated with an oxygen sensor. The oxygen sensor will be useful not only to monitor breathing in intensive care units, but also to check that packages of food, or containers of semiconductors stored under nitrogen gas, remain airtight. The oxygen sensing chip sends light pulses out into the body. The light is absorbed to varying extents, depending on how much oxygen is being carried in the blood, and the chip detects the light that is left. The rushes of blood pumped by the heart are also detected, so the same chip is a pulse monitor. Illustrative example. These are some of the companies offering solutions and offerings for biochips. Agilent Technologies, Incorporated. Biorad Laboratories, Incorporated. Cepheid Incorporated. Fluid Incorporation. GE Healthcare. Hoffman La Roche Limited. Illumina, Incorporated. Osimum Biosolutions Limited.